among the low and middle income countries. China actually has the lowest vaccination coverage in HIV, PCV, Rota, and HPV. So you can see respectively 35%, 10%, 12%, and 1% for HIV, PCV, Rota, and HPV. The lowest coverage lies with the HPV vaccine. In China, only 1%. For Uganda, it is even more than 99%. So we would like to call on the whole society that there should be more coverage. At the moment, there are still questions and doubts or concerns about the effectiveness of vaccines. We have the so-called vaccine hesitancy, and this is the major cause for the inadequate coverage of vaccination, especially in countries like China. Let's take the HPV vaccination as an example. The girls and female teenagers have a very low coverage in terms of HPV vaccination, and this will severely affect the protection. So we need to make more efforts in this regard. The global strategy is to leave no one behind for vaccination. And this is the Immunization Agenda 2030. So for the future, we should fully promote the vaccination programs. For vaccines like COVID-19, we need to promote them. And also, when the COVID-19 vaccination accomplishes a broad coverage, we should pay more attention to the vaccination of other vaccines. So by using the vaccines, we can be faster than the spread of the virus. We can prevent these vaccine-preventable diseases by using the vaccines. So for COVID-19 or for other vaccine-preventable diseases, vaccination is the best solution and the most powerful weapon. I'll stop here. Thank you. Thank you, Professor Zhang. Thank you for the outstanding presentation, sharing with us his research findings. And also, Professor Zhang touches upon important elements like we need very strong public health measures as well as vaccination. And also he emphasizes the importance of vaccines. He convinces us that vaccination is very conducive to the fight against the COVID-19 as well as other vaccine-preventable diseases. Personally, I got into contact with many international scholars and experts. And one important message for me is vaccination is so important. I just want to share with our audience that a few days ago, together with Professor Zhang's team, also made a study and demonstrated the importance of vaccination. Last but not least, may I give the floor to Dr. Linda Stewart, the Deputy Director of Vaccines and Host Pathogen Biology of the Bill and Melinda Gates Foundation. Dr. Stewart is unable to attend the forum in person because of the COVID-19, so I'm going to play her video presentation.
Hello, my name is Linda Stewart. I work at the Bill and Melinda Gates Foundation, where I've been leading the vaccine and COVID vaccine response team. And in normal times, I work on vaccine and biologics discovery. And what I wanted to talk to you about today is acceleration of COVID vaccine R&D and the work the foundation has done to support that. So I will cover today a little bit about the Gates Foundation response and tell you a little bit about 2020 and how over the past year vaccine development was accelerated. And then I will spend some time talking more about mRNA vaccines. So just as background, this is some background on the Bill and Melinda Gates Foundation. We are a large philanthropic foundation. We spend about $5 billion a year across various programs, many of which are in global health and global development, where we spend the majority of our funds and global growth and opportunity. We also have smaller programs we're investing in US education. But for today's talk, really, we'll be focusing on the work in global health and global development in COVID response. In global health, our mission really is to harness advances in science and technology to save lives in developing countries. We work with partners to deliver proven tools such as vaccines, drugs, and diagnostics, as well as breaking new ground and making new solutions that are affordable and reliable. We feel that innovation is very important for health interventions, and we invest very heavily in vaccines to prevent infectious diseases, as well as work on improving growth for family planning, nutrition, and maternal health. In the global health, there are a number of disease areas that we focus on, as shown here: enteric diseases, diarrheal, HIV, malaria, neglected tropical diseases, pneumonia, and maternal neonatal child health. We also are arranged in functional teams that have more technical focus, such as discovery and translational sciences, innovative technology solutions, integrated development, vaccine development and surveillance, and integrated delivery. I sit in discovery and translational sciences, whose goal is to identify, support, and shape scientific research to help it have the most impact to accelerate discoveries and solutions to improve people's lives. This is just a slide that shows the foundation's COVID-19 response, which was starting in January of last year, which was focused on 4 main goals. The first was to protect the most vulnerable by rapidly strengthening capacity and coordination to detect and respond to COVID-19 in key low and middle income and poor countries. We also set out to accelerate detection and containment of the virus by improving capacity for rapid action and surveillance. We also worked on trying to minimize the societal and economic impact of the negative effect of the pandemic on poor countries. And then the fourth was to develop treatments and vaccines for COVID-19. And that's where I will focus on today. In our R&D portfolio, we had 3 main areas of focus: to improve diagnostics, so our test programs, to improve and identify better therapies, so the TREAT program, and then the prevention program, which was to identify vaccines, particularly vaccines that could be scaled and used and affordable for low and middle income countries. So as I mentioned, the foundation has a very long history and interest in vaccines, which is a priority for us. In our team alone, we invest heavily in vaccines, as they've been recognized as being the most important public health tool with the greatest impact. Over the past 20 years, the foundation has spent billions of dollars on developing and delivering vaccines to low and middle income countries with partners such as Gavi. In addition, in looking at the discovery, research and discovery team at the foundation where I sit, we spend about $50 million a year on tools and technologies for the discovery and translation of new vaccines and antibody therapies for global health. In our team, we're particularly interested in, we work on disease-specific areas, but we're particularly interested in investing in a disease-agnostic way and have built a comprehensive toolkit for vaccines and antibody R&D. We build an enabling framework that can support vaccines for any and all foundation priorities. We prioritize cross-cutting platforms, approaches over disease-specific solutions, and we particularly bet on novel technologies and innovations. And we're very comfortable in working on programs with long timelines, such as mRNA vaccines, and we were early investors in the mRNA vaccine space, with our earliest investment being with CureVac in 2013-2014 time. So we've been working in this type of novel vaccine technology for nearly 7 years. So we have focused in our response at really using a 6-step process, really thinking about how understanding the basics of the virus, using existing vaccine platforms, leveraging our network to build an enabling framework, prioritizing vaccines that can be made at a 1 billion dose scale, accelerating their development, and also focusing on their delivery to poor countries. So what happened in 2020 that made vaccine R&D different than in previous times? And how has it been possible really to bring forward so many new ways of and new vaccines in such a short timeline? So normally, as we all know, vaccine R&D is a long process, maybe between around 10 to 15 years. The fastest vaccine previously to be licensed was 4 years, and that was a mumps vaccine. And normally what happens is that these multiple stages of vaccine development, the discovery phase, the phase 1, phase 2, phase 3, and registration phase, all happen in series, one after the other. And then later on, as we know, get closer to thinking that the vaccine is going to work, only then do you begin scaling up and manufacturing. But what has happened over the past year really was a great shortening of many of these stages to really concatenate that whole process down to a 1 or 2 year period. And one of the things that has been significantly shortened was the discovery and preclinical development period. And I'll explain a little bit, that's where I'll focus on today, to explain what was done differently there. But all the other phases, although they were shortened, they still happened in the order that they normally happen: phase 1, phase 2, phase 3, and regulatory process, because that is what is necessary to ensure that a vaccine is safe and efficacious. So nothing has been shortened in those normal safety processes and efficacy studies that are required to bring forward registration. The other thing that has happened is that people began manufacturing much sooner before they even knew that the vaccine was working. And this has helped speed up the process. But manufacturing remains a bottleneck for delivering the number of doses that we really need to be able to protect everybody on the planet. But what I'd like to focus on now is what changed in the discovery and preclinical development. So normally this is a process that can take a very long time. The probability of success of something this early on is very low. It's about 5 to 10%. But what we did differently was we really focused on using novel platforms such as mRNA. And there was a very large portfolio of candidates that were brought forward simultaneously. A number of partners were across the globe have been involved in this, and we have worked extensively at the foundation with our partners CEPI, who are also partners to the Ministry of Science and Technology in China, as well as the National Institutes of Health here in the US. So normally vaccine, the way of thinking about a vaccine and vaccine development is shown here. So often we will take information about what happens when people are infected with a pathogen and try and understand what is the protective mechanism. Is it an antibody? Is it a T cell? Normally we look for antibody mediated mechanisms because these are easiest to drive through vaccination. Once we find the antibodies that protect, we try and understand which antigen in the virus elicits them. So in the case of a virus such as a coronavirus, are those protective antibodies to the spike protein, to the N protein or the M protein? But that's something that is, that's basic work that needs to be done for us to know which antigen to use in the vaccine. Once we know what antigen to use, we then start working with the antigen to work out how best to structure it, how best to stabilize it, what to do with the antigen. This is an antigen shown here, sorry, looking at an HIV antigen just as an example, but a similar work for coronaviruses. Those antigens are then designed through structural biologists and then put into animal models for testing. And once we've identified that normal testing through that preclinical testing, they go into healthy volunteers to be evaluated and then put into the phase 1 and phase 2 studies. But what happened differently in COVID-19 was this. So in COVID-19, there actually had been a lot of foundational work that had been done on the coronaviruses, particularly by the National Institutes of Health, with a group by Barney Graham, who had been working on SARS and MERS as prototypes of the coronavirus family. They had done a lot of work looking at the structural biology of the coronavirus spike protein. They knew that it was a good target for neutralizing antibodies and could be a good vaccine candidate. And all of this work had really been laid, the ground had been laid by the time over 2017, 2018, 2019. By the time SARS-CoV-2 came along, the crystal structure was very rapidly solved based on the precedent of the previous coronaviruses. The immunogen was designed, and this used to stabilize two prolines had been added to improve the immunogen. And this is the immunogen that is in the large majority of the vaccine candidates that we have currently seen. So Moderna, Pfizer, BioNTech, and Novavax all use the same stabilizing strategy. These were then used, there was a decision made to use vaccine platforms, and we'll talk about those in a minute, and then very rapidly go into preclinical and animal models, such that the actual human studies began within 3 months of identification of the virus. So focusing more on the de-risk platforms, clearly there was the mRNA, and we'll come back to that, but 2 additional approaches were used, of course. So the viral vectors, such as the chimpad and CanSino would be a great example of this, the adenovirus vaccines, protein subunit vaccines, such as the work going on in China with Warvax and Clover as examples, and then the mRNA vaccines, which I will talk more about. I haven't included in here the live viral live or inactivated viral vaccine family, just because those are very well known to the audience, and China really is a leading player in that, and I don't think I have anything specific to add to that, that knowledge set. But focusing on mRNA vaccines, I think there really have been 3 companies, Moderna, BioNTech, and probably hopefully CureVac, that have led the way in this. So I'll talk a little bit more about the mRNA, and I'll really compare them to the subunits, because in both of these cases, these are really striving to use proteins, single proteins from the virus to elicit an immune response. So subunit vaccines are really proteins that are generated through DNA and RNA cloning. They're expressed in cells, and the protein is purified and then formulated into a vaccine that's then administered to people. The purification is often very complex. The manufacturing process is often very difficult, and it must be delivered with an adjuvant to elicit an immune response. There are some advantages. It's a precedented approach, so we know a lot about subunit vaccines. The disadvantages, however, are the manufacturing is complex, requires a different process for each antigen, can be time consuming, and the scalability is unpredictable. And the adjuvants themselves also add complexity because they add to the cost, they can be limited in supply, and the safety is unpredictable. mRNA vaccines are, however, different in that what you do is you add the mRNA to a lipid nanoparticle carrier, and in this formulated mixture is then made into the vaccine. That vaccine is given to people, and the mRNA is then, once inside the body, translated into protein to elicit an immune response. So the mRNA is formulated with a lipid nanoparticle. The mRNA is then injected, and once it's inside, it is translated into protein within the vaccinee. And the LNP and the RNA self-adjuvant to elicit the response. The advantage of the mRNA is that it's very quick. These mRNA vaccines have been licensed within 10 months. It's very flexible. The antigen is just determined by the mRNA sequence, and so it can be swapped, changed very easily, such as for the variants of concern. The manufacturing process is the same for any antigen. We didn't know before the pandemic whether it was scalable, but we do know now that we can achieve billion dose scale. And we now have a safety dossier, both in all age groups, even in pregnant women and also children down to the age of 2. So the disadvantages, of course, is this is novel. These are the first licensed mRNA vaccines, and we still don't know the long-term safety, but there is nothing in the short-term safety to make us concerned. So to explain the mRNA vaccine principles and production in a little bit more detail, here is just a cartoon of mRNA. So the sequence encodes the sequence of the protein to be expressed. The sequence can be optimized for expression, so you can change the use of the codons to improve the amount of protein that's expressed. The mRNA is produced in vitro by in vitro translation in E. coli. Modifications are made to the mRNA to make it less inflammatory, so you can use unmodified or modified mRNA. So unmodified, CureVac uses unmodified, BioNTech and Mrna use modified mRNA that detune the inflammatory response. And then the mRNA undergoes capping and additional modifications, so it's ready for translation once it's delivered into the cell. The mRNA is then formulated into a delivery package called a lipid nanoparticle, and the LNP is used to encapsulate the mRNA to protect the mRNA from degradation. It increases the mRNA uptake into cells, and it is required for protein, the delivery of the mRNA is required for protein expression. LNPs also stimulate the immune response and act as an adjuvant, and they can increase the efficacy of an mRNA vaccine by about 1,000-fold. The mRNA is manufactured by really the co-formulation of these two. The manufacturing process is more complicated than manufacturing a small molecule, but it is certainly easier than manufacturing other biologics such as standard vaccines or antibodies. Once the process is established, it is the same for any mRNA. We know it's scalable. The manufacturing footprint is relatively small, meaning that there is a very low capex. So what do we know now that we have mRNA vaccines, and why are we particularly excited about them? I think what we know is that having de-risked them with COVID-19, we know we can deliver mRNA and LNP safely to people. This means that there are many opportunities now available to us for new mRNA vaccines, be they HIV, TB, malaria, diarrheal diseases, flu, or so on. So many opportunities for new vaccines, and of course, for future pandemics if we would need them. The safety database in pregnant women means that we could use them for maternal immunization, And there's also data showing that we can combine mRNA vaccines to allow us to do combination vaccines. So for instance, we could do flu with coronavirus all in a single shot. But there are additional things that are now possible with this. We can do different things to improve the RNA. So we can actually make the RNA more stable and make more efficacious products that will improve the applicability and deliverability by improving thermostability and potency of the mRNA itself. And then we can also deliver other RNA-based therapies. So we can use RNA now, we can start exploring using it for gene editing or gene delivery, such as antibody expression, or the delivery of regulatory RNA, such as siRNA therapies, or as I said, for gene editing through CRISPR-Cas programs. The second thing that we can do now is that we know the lipid nanoparticles can deliver mRNA into the cells in humans. These can actually be improved. So we can improve these to change the biodistribution and where they go in the cell and in the body, such targeting particular tissues, or we can use different types of delivery mechanisms that may have different advantages, such as thermostability, cost, and scale. And then we can also do additional tweaks to the delivery system to really improve the LNPs, get tissue-specific expression, or even deliver larger cargo or multiple cargo at once to a particular cell type. So I think I will end here by just saying, I think the de-risking of mRNA vaccines has opened up not only opportunities for mRNA vaccines, but also for a world of opportunity of RNA-based therapies. And we're particularly interested in applying those to global health. So with that, I'll say thank you very much for the invitation to come and talk, and I hope you enjoy the rest of the meeting.
Thank you, Dr. Linda Stewart, for the wonderful presentation. It's very detailed and very comprehensive. Let me try to summarize her presentation. As we all know, the Gates Foundation has invested heavily on the vaccine development and also in global health over the past 20 years. Dr. Stewart has shared with us the initiatives and the investment made by the Gates Foundation in global health, in particular in vaccine. She explained to us why it is so expensive to develop vaccines, and also she shared with us the great significance of the mRNA vaccine technology. It is truly a new opportunity. Also, the mRNA technology can be expanded for its use or its application in treating the diseases. So we can have the RNA-based therapeutic technologies. Indeed, it's a promising technology that will bring health and happiness to the mankind. I'd like to say thank you to all of our 4 keynote speakers. As we all know, this Global Health and Development Forum is part of the 2021 Pujiang Innovation Forum. And the forum attaches great importance to the experience of scientific and technological innovation and international cooperation. In the following session, we will have 4 guests who will share their experience and the results. As invited by the Gates Foundation, please welcome Professor Zhou Xiaonong, who is currently the director of the National Institute for Parasitic Disease, the Chinese Center for Disease Control and Prevention, and also the Deputy Director of the School of Global Health, School of Medicine in Shanghai Jiao Tong University. Let's welcome Professor Zhou. Thank you. Thank you for the kind invitation. I will briefly talk about the China-Tanzania project on malaria control. As you all know, malaria is a disease transmitted by mosquitoes. It poses a high or a heavy disease burden globally. In 2019, according to WHO report, there were an estimated 229 million cases of malaria. The estimated number of malaria deaths stood at 409,000. It is said that for every 2 minutes, there is a child under 5 years old who died because of malaria. So malaria is one of the 3 major diseases in the world. That's why globally we have the Global Fund against malaria, TB, and HIV/AIDS. Domestically, in China, we also find a lot of attention and support to the fight against malaria. China has achieved a lot in malaria elimination. Here you can see that prior to the year 1980, the prevalence of malaria in China is about 100 per 10,000, quite similar to the situation in Africa. However, over the past 20 years, we can see the rapid drop of the prevalence of malaria in China. And China now proposed to eliminate malaria. So here you can see that starting from 2010, the prevalence of malaria in China was greatly reduced as we adopt a strengthened monitoring and response measure. In the year 2014, malaria cases were mainly found in the border areas. And in the year 2016, we had the last case of malaria reported. And since then, there were no malaria cases in China reported. So based on WHO criteria, for 3 years in a row, 0 malaria case, and that's why we were declared as, or we submitted the application to WHO as a malaria-free state. WHO is now reviewing our application, and it is likely that this year WHO will announce China as a malaria-free country. Let me share with you the lessons we have summarized in the fight against malaria. Lesson 1, government commitment. Lesson 2, phase and strata-specific strategy. In particular, in the early days, we have the comprehensive strategy, and later we have the strata-specific strategy, and then we have the quick identification and source tracing. So gradually, prevalence was reduced. Lesson 3 is the evidence-based approach and innovation. We had the scientific studies to provide the policy input. And lesson 4 is the capacity building. The whole disease control system, the clinical system have both been well trained to improve the capacity. Lesson 5, sustainable financial support. Starting from 2003 to 2012, Global Fund provided 320 million to China. China became a major recipient country. And then in 2010, we launched the malaria elimination program. So the fund from Global Fund is really important. And lesson 6, management of high-risk region and population. Here you can see that in the border region, apart from the 3 lines of control domestically, we also had the cross-border cooperation. We worked together with our Myanmar counterparts to reduce the inbound transmission of malaria. And lesson 7, international cooperation. Just as I have mentioned, we receive international funding, and also we had the collaboration with WHO. Furthermore, as a recipient country, we begin to cooperate with relevant countries and organizations so that in the future we are also capable of sharing our experience and providing technical assistance. As you know, Professor Tu Youyou received the Nobel Prize in Physiology or Medicine in the year 2015 because of the anti-malaria active product of Artemisia. The Chinese method has also won approval and recognition from WHO, and the Chinese approach has been included in the WHO technical guideline.
After we initiated malaria elimination, we began to talk about how to cooperate with African colleagues, because with the number of cases getting less and less, close to zero, finally to zero, there are still a lot of imported cases. Every year, about 1,600 to 3,000 imported cases, out of which 96% are from Africa. So if we are to help African colleagues to control malaria, we are not just helping with malaria control in Africa, but also helping reduce incidence of imported malaria cases in China. This will also, if we do not do so, it will become a time bomb for malaria elimination in China, because we have mosquitoes in China. So if we have imported cases with mosquito, the disease still can spread in China. Therefore, we have established an alliance or network for cooperation with Africa. And such work is also quite helpful in Africa. And the WHO has also assessed the situation of malaria across the globe, in particular in Africa. You can see since 1980s, the incidence rate of malaria in Africa has greatly reduced. And that is something that we need to study further. And the key is how to share China's experience with Africa. And how to get the same results. Like in China, for example, with more than 30 countries in Africa, we have established about 30 anti-malaria centers. But after the establishment of the centers, if you do not follow up them, the centers were just established and not effective. That is also a lesson for us. We can see that in different time periods, the malaria control work in China changed from eliminating malaria in China to gradually become focusing on preventing imported cases. And to prevent imported cases, we use 137 monitoring approach. That is to say, the case should be reported within 1 day, and within 3 days, the case investigation should be completed, and within 7 days, we should get clear about the source. We cooperated with Tanzania, which started in 2002 with the help of the WHO. Together with African colleagues, we had some discussions on how to land the Chinese practices in Africa. I mentioned 137 monitoring plan, and this has been recognized by the WHO. Then how to do it in Africa? We received DFID assistance of the UK. We have been doing such cooperation for 3 years. You can see across the whole country of Tanzania, malaria prevalence in east and the west is high, but the middle region is the low prevalence region. So our strategy is to focus on the key areas. We selected a small area for phase 1, and we hope that we can have phase 1, phase 2, and phase 3 cooperation. You can see for phase 1, our objective is to establish partnership to translate Chinese experience in malaria control to locally tailored strategies in Tanzania. Of course, we selected the 2 areas in red as the intervention areas. The green areas are reference areas. Total population is about 200,000 people. When we designed the cooperation, the WHO proposed 3 T: test, treat, and track. Diagnose, treat, and track. Test, treat, and track. But it didn't give a timetable. So the contribution by China is the 137 model. But if we directly use it in Tanzania, it doesn't make sense, especially reporting of all cases within 1 day across the country. They don't have the IT system to support such reporting. They are doing the reporting manually using the reporting cards. It will be even difficult to get all the cards within 3 months. Normally, they can complete it within 6 months. So if in a manual way, it is very hard to block the spread of the disease. So we discussed with the related experts. We think that we can hire the local barefoot doctors. We call them volunteers. We train the volunteers, and every day the volunteers in the communities, in the villages will conduct monitoring. Once a case is detected, they will use the fast test reagents to quickly diagnose. Every day they have a pooling of the case information. If an area with severe infections with a severe situation is emerging, then we will use comprehensive chemotherapy to deal with it. So this is the phase 1 strategy. That is a strategy of enhancing treatment and monitoring. And this strategy has been published in the magazine. In our work, we focus on this strategy and carried out the following work: community mobilization, better educated villagers, cost effectiveness and practicality of entomological research, and assessment. We have also invited the WHO experts to conduct the assessment as a third party. Here are some data. You can see for severely hit area, incidence rate was reduced by 85%, and for those with a better situation, the incidence rate reduced by 70%. That is active surveillance. And for passive surveillance, that is in the data collected from the local health system, also indicates a decline of epidemic in those areas. So that is the phase 1 results. And the WHO experts conducted external evaluation. They spoke highly of our practices, saying that we used a very cost-effective way to quickly reduce the incidence rate. They also asked us to keep on the good work. And the Gates Foundation also was involved in the evaluation, and they also think that we should roll out such work to a larger area, and they will also evaluate after it is expanded. So this is Chinese experts working together with Tanzania experts, and the evaluation team of the WHO spoke highly of our work here. We visited Switzerland. Chinese ambassador, Swiss ambassador, and German ambassador all attended the stock-taking meeting and talked about their hopes for our future work. So we are now conducting phase 2. Under the support of Gates Foundation, we'll conduct the evaluation at the end of this year. But the project has also been affected by COVID-19. The local areas were hit by both diseases. But in these areas, compared with other areas, the incidence rate of malaria is very low. We have also expanded the pilot. Originally 200,000 people, now close to 700,000 or 800,000 people in communities are participating in our project. We hope that we can integrate this strategy into the local national malaria control strategy. And we are now engaging with the Ministry of Health of Tanzania for discussions. In this process, we have also combined our application research with the analysis of the local vectors such as mosquitoes. Cooperation mechanism is the key. In Tanzania, they have established a working group. On the Chinese side, we also established a working group. Often, we have joint meetings to have discussions. In the long run, we will also have phase 3 for scaling up. Currently, the WHO has offered support for us. We have secured Peace Fund of the UN to do it in 4 countries, Tanzania, Zambia, Burkina Faso, and Senegal. To scale up in the 4 countries, we hope that the yellow part and red part can further shrink, so that the malaria cases in Tanzania can be further reduced. This project has also received a lot of attention from Minister Li Bin of the NHC of China and also Academician Gao Fu. We are expecting these outputs by the end of the year, which is shown on the screen. And we hope that our work in the 4 countries mentioned can be further strengthened. We would like to acknowledge the Chinese government and multilateral international partners, including DFID, Gates Foundation, the World Health Organization, and the Global Fund is also prepared to participate. In particular, I would like to highlight that the local African colleagues have made a lot of efforts and offered great support for this project. We hope that through this project, China Wisdom and China Model can also be landed in Africa. I hope that the audience, especially the young colleagues present today, can also participate in such a project. And malaria is a very good area for you to participate. I'm looking forward to your attention and support. Thank you. Thank you, Professor Zhou, for your wonderful presentation. You shared with us the malaria elimination experience in China, including 137 model, and you shared with us the international cooperation experience. For example, the sharing of China experience in Tanzania with good results. We are looking forward to more results coming out. Without international cooperation, it will be even harder to cope with mankind's challenges. Thank you. Next, I would like to invite Professor Jiang Biao to make a presentation. He is a doctoral student and researcher jointly trained by London University and Shanghai Institute of Organic Sciences, Chinese Academy of Sciences, and once served as a visiting scholar of DuPont pharmaceutical company in the United States. He also worked in the Bangkok Innovation Center, not for research, but for the translation of China experience in Southeast Asia through Bangkok Innovation Cooperation Center, Chinese Academy of Sciences. And he will make a presentation on drug development for vector-borne infectious diseases prevention and control based on Chinese drug sources. He's also a close friend of mine. Let's welcome him to the stage. Thank you, Professor Cao, for your introduction. I would like to thank Pujiang Innovation Forum Organizing Committee and the Bill and Melinda Gates Foundation for the invitation. With the funding of Gates Foundation, we have conducted a project that is novel vector control based for vector control. I'm going to skip the background as Professor Zhou has already elaborated on the background, in particular the WHO report. So actually, for every minute, that means 60 seconds, there would be an African child died of malaria, and every year more than 50,000 people died because of malaria. Over 90% of the deaths occurred in Africa. Malaria is a disease caused by Plasmodium, which is transmitted to humans by mosquitoes that carry the parasite. Parasite Plasmodium will cause the malaria disease. For every minute, I'll try to see the slide and move on the slides. So we can see that malaria in China has been disappeared for 4 years. But this is the 2017 figure, and you can see that in Africa, the color is quite dark. So there's still a daunting challenge to control malaria. China did quite well in malaria control. When we applied for the project, we also tried to share the Chinese experience. We use biological
mosquito larvicides and also the long-lasting insecticide-treated nets, bed nets, to protect the people. For low malaria intensity, we use spraying and also the larvicides. And malaria control is a national strategy. From 1949 to 1959, at that time, the cause of the transmission was not known. And then we have the outbreak in the 1960 to 1979. Later, we have the artemisinin developed, and this is the characterization of artemisinin. This is headed by Professor Zhou Weishan and Professor Tu Youyou, the Nobel Prize winner, extracted artemisinin from the Chinese herb plant. And with artemisinin, thousands and thousands of lives were saved. So Professor Tu received the Nobel Prize in the year 2015. This slide is about mosquito. Mosquito is a major vector for malaria. Every year, mosquito claimed on average 830,000 lives. Mosquitoes can cause many diseases like dengue fever and the yellow fever. And malaria was a major disease caused by mosquitoes. Here you can see over 50% of the world population are threatened by the vector-borne diseases. So if you travel to Africa, you are strongly suggested to take preventive measures against such mosquito-borne diseases. How can we effectively control the spread of malaria or other vector-borne diseases? We need to do further studies, even though internationally we have already applied more than 17 types of insecticides or pesticides. This slide is about the Anopheles sinensis. It is still widely distributed in East, Southeast, and South Asia. It's a vector of malaria and filariasis. Then you can see the Aedes mosquito. Some of the Aedes mosquito are carriers of arbovirus diseases such as yellow fever and dengue. Two approaches have proved highly effective in controlling malaria vectors. That is the indoor residue spraying and the use of insecticide-treated nets, just as Professor Zhou has mentioned. Up to now, WHO has approved 13 pesticides or insecticides. However, with the long-term use, we can find some drug resistance in these pesticides. And also, the mosquitoes developed resistance to several insecticides, including pyrosoids, organophosphates, carbamates, DDT, and etc. That's why we need to develop new drugs against the malaria, the disease. In particular, we need to develop new pesticides to get rid of the toxin caused or transmitted by the Aedes mosquito. So we try to use the TCM to develop new tools. And here you can find the project that we have had. We wish to develop novel, efficient vector control tools originating from TCM. As early as in the 1100s, the Chinese poet Lu You wrote a poem about the mosquitoes and the way to get rid of the mosquitoes. For our project, actually, we enjoy a very solid basis, as there is a database developed by the Chinese Academy of Sciences, in particular the Shanghai Scientific and Technological publishers. This TCM database has collected information about herbs and the compositions. Altogether, about 300,000 pieces of TCM data are compiled in this database. So in our institute, we enjoy such a good platform. For example, if you click into the term Hanqing, you will find the detailed introduction and the composites this TCM herb has. So this is the workflow on the platform. With this computer software, we are able to develop the potential pesticide. With this platform, the automatic screening and selection of the TCM herb or composites can be realized. This can greatly increase the effectiveness and reduce the laboring or the time and efforts. The manual efforts can be greatly reduced by using this platform. So this is a sort of artificial intelligence. So we will have the prediction first. If the result is quite good, we will then move on to do the synthesis and then test. If the test result is not as good as we have expected, we can come back to modify the composition. So with artificial intelligence and the chemistry, we can improve the efficiency of identifying the active ingredients. We have applied this approach in the development of new composites or new drugs. This is for the malaria. So we put them together. This is for TB, the anti-TB compounds. We had the natural ingredient from a garlic. So we can come up with a very effective compound or an anti-TB compound. We are working on this project, and for the next phase, we will continue our collaboration with China CDC. So we can select some properties and help to develop potential pesticides. Now you can find more than 2,000 pesticides in our database. We can screen the database and we can control the molecular weight, say 200 to 500, and for log P from 3.5 to 6. And for the hydrogen bond, the donors we can determine, or we can control the quantity and can do the screening. Let me share with you some examples. In China, we have a lot of TCM prescriptions which are collected and printed. Take Aicao as an example. We can find this TCM prescription. This is the Folium Artemisia AG. With a burning, we can get the effective element. And also, we have another example of Magnolia officinalis.
We can find Magnolia officinalis and Folium Artemisia AG in our database. The former has 42 compounds. The latter has 367 compounds, totally 409 compounds. Through the screening by the computer, there are some recommended and validated numbers of compounds. For Magnolia officinalis, 15. For Folium Artemisia AG, 123, totally 138. For Magnolia officinalis, we conducted screening of the compounds. If you use, we use the pyrozoid as comparison, we found the data are very consistent. And if we use protein, to do the docking, we found the bonding rate is also very high. So there are 3 types of amino acids and protein for docking. If it is 10 to 14 or 8 to 14, then it is all effective. And we have also designed the following line of compounds to calculate back to see whether these compounds are stronger than the upper ones. If yes, then we will take them for synthesis and manufacturing. For Magnolia officinalis, we identified around 20 artificial derivatives, very good ones. And for Folium Artemisia AG, mainly brass ones, for example, 115, 34, 44, very effective. But they cannot be made into pesticides or insecticides. You can see for those that are made into insecticides, the value is 0, because for these compounds, they cannot be volatile. That is to say, to drive mosquitoes, they must be burned so that the smell, the compounds, can become vaporized to drive away the mosquitoes. That proves the method I talked about previously. The method used by ancient people is effective but cannot kill the mosquitoes. They can just drive away the mosquitoes. Through the computer screening and recommendation, we need to do deeper testing. This is the WHO prescribed procedure. That is to say, the activity must be topically active or lethal against adult mosquitoes. And about the speed of effect, it must have lethality within 72 hours of a 30-minute exposure. And the potency must be at least equivalent to that of permethrin, or at least equivalent. Another indicator is cross-resistance. It must be active against both target site and metabolically resistant mosquito strains. So every time we use 30 mosquitoes for the testing, and we must have more than 70% lethality. For every 0.1 milliliter, at least the mosquitoes, 70% of the mosquitoes must die. If 100%, that would be even better. So we conducted the bioassay for all the effective compounds. You can see that for 1 hour knockdown rate is 100%, and 24 hours mortality is also 100%. So you can see at least the equivalent to that of permethrin. And now let's look at the prediction. And the bioassay, you can see the 3 lines in yellow are still 100%, but the one SIOC 20123-1-1 is the one that can be used for repetitive testing to get more fundamental mechanism of pharmacology and toxicity. So we used computers to do the prediction, and it is in line with actual results. For number 2, it can be 100% as a pesticide. I mean, the SIOC 202012312. So then, theoretically, we try to see whether it is in line with previous literature. This is in the protein space. We are testing the angiotensin converting effect, the interaction between plasmodium, interaction between ligand and targets. This is the electron density. To kill the mosquitoes, plasmodium, there are 3 proteins. One is the acetylcholinesterase catalytic subunits from Anaphylis gambiae. You can see 5 compounds are in the upper part, both in electrostatic potential and also in lipophilic potential. Such studies are very important for insecticides. After 1 year study, we established a method And we also use the computer screening platform. In the TCM database, we screened 25,600 compounds, and after computer simulation, we screened out. We got 388, and then we did bioassay, and then we further get a short list of 33 compounds, and then we conducted testing in CDC. Now we have 11 plus 9, totally 20 compounds that are possibly potential to become the next generation new pest insecticides to drive away mosquitoes. To kill the mosquitoes, we then draw the conclusions. For the compounds, if it is controlled between 200 and 300, it is effective, and the log P is 96 or 97. They can be used as mosquito killers. So that is a very good validation of our method. This has established a technical support for the discovery of lead compounds. We have also developed an information inquiry system for lead compounds. Our research institute conducts the synthesis, computation, and design. It also involves chemical research, and the CDC helped us with technical assistance for disease control and prevention, like the bioassay and protein research, and Lancet connected us with LCC of Europe to fulfill the mission of eradicating this disease and to develop products in sync with WHO's malaria prevention strategy. By utilizing such a platform, we can make sure of effective prevention and control that is also environment friendly. We used this platform to realize the study of mosquito killing and driving away mosquitoes. In 2020, we got a new Anopheles insecticide that can kill the mosquitoes that are drug resistant. In the first half of this year, we conducted testing in CDC. We were lucky to have obtained several very active compounds with very low toxicity. And after the testing, we made further improvements or screen or test new compounds. In the next round of testing, we will finally identify the compounds, and then we will conduct the toxicology study. We have 3 teams. One is our institute, led by me, and the second is Chinese CDC, led by academician Fu Gao, and Dr. Duan and Dr. Wang, and the third team is led by Danny Lin of Lancet Group, together with Bill and Melinda Gates Foundation and IVCC. They are in charge of the future QPS development. This is a picture taken in 2019 in Sydney with IVCC. We were sharing with us our working procedure. And I would like to thank Bill and Melinda Gates Foundation for the support for this program. I hope that through this program we can thoroughly kill the mosquitoes and make our contributions to malaria eradication. Although China is polio-free now, but there are still a lot of imported cases every year. It's like that such cases are not happening around us, but actually they are in the form of imported cases. So we hope that malaria can be eradicated in Africa and then in the whole world, so that we can have a better planet Earth. Thank you. Thank you, Professor Zhang. Thank you, Professor Zhang Biao, for your very scientific presentation to drive the animals to more than 10,000 miles away. We hope that through the team effort of Professor Zhang, through the combination of efforts between the industry, the academic research institutes, and academic community, we can make further contributions to malaria eradication. The next presentation will be made by China representative Madam Yuan Yuan of PAS. He will share with us past work in China to advance global health collaboration. Professor Yuan, you have the floor. Good afternoon. It is a great honor to share with you past work here, and I hope that through sharing with you the work experience of past, we can also offer some inspirations for future innovations and cooperation. Since we are behind the schedule, I will finish my presentation very quickly. PAS is a non-profit international organization. We focus on 5 platforms, including vaccines, diagnostics, drugs, devices, and health systems. Now, globally, we are a global team of 1,500 plus people working in 70 plus countries. Although we are headquartered in the US, but in recent years, one of our work priorities is to put more of our talents and resources in developing countries, including Asian countries and African countries, including China. But China holds a special position because starting from 2000, China gradually ceased to be a recipient country. So we deem China a very important partner. Just now, Professor Zhou mentioned that China has a lot of experience in health systems in malaria control, which is rolled out in Africa. We can see that the other 4 platforms of PAS are products, and China boasts great potentials in those 4 areas.
那举个简单的例子，从疟疾来说的话，是全方位的一种工作，包括非洲和亚洲的疾病防控，然后也包括一些工具的应用，比方说诊断试剂，还有疫苗。 对于新发的传染病，我们也是非常重视。其实，在新冠疫情发生之前，也是和前面几位专家谈到的一样，感觉到人类是会面临大流行或者是局部的爆发，所以我们有一个叫全球健康保障的项目，去帮助这些国家的卫生系统建立好这样的体系，然后在疾病到来的时候能够有所防范。 另外一方面，在新冠的过程当中，我们现在在中国做的大部分的工作是帮助企业。
In COVID-19, we are helping the companies so that their products can be enlisted in the WHO procurement list. And here you can see the idea. We come up with an idea, then we have potential solutions. We devise and test the solutions over and over and navigate hurdles to acceptance and use. Then we support the countries and the communities in adopting the solutions. And also we collect the data. Then we scale massively to meet global needs. So the ultimate purpose of innovation is to have a widespread access to the solutions and achieve impacts. PAS was set up in 1977. Since then, we are looking for the global health innovations. We notice that At first, the innovations generated in developed world, but now we found the reverse trend, that is, innovation originates from the developing countries, and China holds a big potential in this regard. For example, for diagnostic and devices, PAS developed a women's condom, and the technology was transferred to Dahua, China, and also we helped Shanghai Dahua to get the WHO PQ. Another example is we worked together with Professor Qiao Youling and developed the HPV testing. The technology is then transferred to a company and got the WHO PQ in the year 2018. Furthermore, we are developing the malaria RDT. We hope that in the near future, these products can be available in the world. As for vaccine cooperation and innovation, this was mainly achieved over the past 20 years with our partner, the Gates Foundation. For meningitis, you can see that it helped to prevent the African outbreak. For Japanese encephalitis, JE, this is also a key vaccine we have had with the cooperation from the Gates Foundation. We hope that such products can be easily accessed. Now let me talk about the reason that PAS gets so actively engaged in vaccine development. Indeed, people usually think that companies take the leading role, but here you can see that for vaccines, there are 4 scenarios of the commercial value of the vaccines. It is quite natural that we have such scenarios. So we may have the uncertain scenario or assistance-dependent scenario. If it is a local outbreak, for example, in SARS, Chinese companies also developed a vaccine, but SARS disappeared. So the investment by the company may be wasted. So companies would have no motivation to develop such products. Then organizations like us can help the companies to overcome the obstacles and financial difficulties. Also, there are countries who are short of funding, and also they cannot provide stable ordering. So organizations like Gavi can help by purchasing in large quantity or placing the order in a large quantity. Companies would be motivated to produce the products. Also, the R&D of vaccine is full of difficulties. As you know, we have the valleys of death. That means the development may not be successful. Also, for vaccines, vaccines are designed usually for the healthy population. Then, without the regulatory approval, it would be difficult to develop and market the vaccines. So we are trying to offer help. Take the rotavirus as an example. We got the technology from the US, and also we set up a platform where the companies can share their data and their experience with other companies. For Chinese companies, even though they are a little bit behind in terms of the R&D, they can take the latecomer advantage, and they can also be actively engaged in the R&D and production of the vaccines. Today, let me focus more on the JE vaccine, just as Dr. Steve Davis has mentioned. For JE, it is no longer so serious in China, but globally, it still poses a severe disease burden, as there's no effective treatment for Japanese encephalitis. In the year 2013, GE vaccine from Sinopharmaceuticals was pre-qualified by WHO. This is data from China CDC showing the impact of JE vaccine. Two vaccines are used. One is the inactivated, and the other is the live attenuated. We choose the live JE vaccine because it only takes 2 doses, while the inactivated JE vaccine requires 4 doses. Actually, there's also the 1-dose regimen available in the world, but it's not widely used. CMBG Chengdu is an experienced manufacturer of vaccine. It took us 8 years to get the WHO PQ. Here you can see the milestones along the 8 years of WHO PQ. The year 2011 marks a significant milestone. That is, the Chinese NRA passed WHO assessment. Only with that can we apply for the PQ of WHO. Then in early 2012, CMBG Chengdu submitted the application to WHO and received the site inspection in the year 2013. Also, we need the talents, the human resources. So the quality management system is indeed an area where we need strong human capacity. Also, for Chinese vaccines to go global, we need to have more talents, like in the clinical trials and establishment of pharmacovigilance and etc. We always need human resources. For the JE project, the Gates Foundation has contributed greatly. You can find the 7 clinical trials, and you can imagine how much the investment is needed. Now with the clinical trials, data is accumulated to convince the regulatory authorities. Now, 12 countries are using the GEV produced by CMBG Chengdu and protecting more than 60,000 children in the world. And every year we provide the prediction volume, the production volume forecast to CNBG Chengdu. And also, even though the project completes, we share this important tool with our partner, the CNBG Chengdu. Also, CNBG Chengdu was not affected by natural disasters like the earthquake in Sichuan province. And also, the business continuity is ensured with our contribution, with our collaboration. PASS has also been very supportive on sustainability. We are aware that projects may be completed, yet we need sustainability for long-term growth. So, when we cooperated with CNBG Chengdu, we helped the company to build the system. So we helped them by establishing this pharmacovigilance system. So our experience gained from the GE vaccine production cooperation have been summarized and also updated. In short, to develop a vaccine, it takes time, but if companies work together with the international partners or counterparts, and if the proper arrangement is made, it will facilitate the technological transfer and the vaccine production. By doing so, the product can increase its competitiveness. Also, we can see that the Chinese government is encouraging the Chinese companies and the vaccines to go global and serve the global health. And this is our Vaccine Innovation and Acquisition Center. We are facilitating the end-to-end vaccine development and delivery. We also look forward to work with domestic partners and accelerate innovation from idea to impact and contribute to global health. That concludes my presentation. Thank you.
Thank you, Madam Yuan Yuan, for your introduction on PASS. I hope that there will be more cooperation between PASS and Chinese enterprises so that they can go global. The last speaker is Professor Yang Yue of Tsinghua University. She is China's first pharmacy management. She is the first doctoral degree holder of pharmacy management direction. And she will give us a presentation on project and progress of Tsinghua University think tank for drug and vaccine regulation. Hello, I'm Yang Yue from Tsinghua University School of Pharmaceutical Sciences, and I will share with you a project supported by Bill and Melinda Gates Foundation, that is
the
Tsinghua University Pharmaceutical and Vaccine Products Regulatory Science Think Tank project. We established a Pharmaceutical Regulatory Science Institute, and Tsinghua University leadership pays a lot of attention to this, and the development of this institute have become a top priority of the university. The School of Pharmaceutical Sciences, as a core college or school for regulatory science, has made a lot of fundamental work in the upfront stage. Because of such a background, Bill and Melinda Gates Foundation offered us a lot of support. In the end of 2019, we signed cooperation agreement for this project. Tsinghua University's pharmaceutical regulatory science position is it is strategic, key, innovative science. It is quite different from the previous pharmaceutical management discipline. It is more about the scientific aspects of regulatory over pharmaceutical products. So first is the problems in regulatory framework and how to address the problems through research. We pay attention to the bottom layer scientific logic to address the problems. And then finally we make recommendations for future pathways for future regulatory work. In the overall regulatory science research, we focus on the strategic key common difficulties, especially those that constrain the product development, the development of the industry, and the regulatory efficiency. You may have this feeling, why that a legal framework has been established, but the regulatory pathway is still very ambiguous, very unclear. It is like walking in a maze. Although you can always reach the destination point, but you need to try different pathways. Not every pathway can lead to the destination. Therefore, in the preliminary stage, in establishing a legal framework, when the supportive legal documents are not developed yet, this will happen. So we are to address this problem. On October the 30th, 2020, we initiated this project. We had this project initiation meeting. We invited leadership from NMPA and experts from universities and the leadership of CDC, in particular, Professor Xu of Bill and Melinda Gates Foundation and experts of the WHO, who all attended our project launch meeting. And then we had several meetings related to the vaccine regulatory science for academic discussions. And for a lot of seminars like this, we reported the phase-by-phase results. For example, the optimization of pharmaceutical products, examination and approval procedure. And the prescription drugs and OTC drugs regulatory pathway research. And NMPA's CDE also asked us to carry out another project, that is pharmacovigilance management norms and adverse reactions surveillance and management measures for pharmaceutical products or medicinal products. Prior to the launch of this project, we also carried out another large project, that is a vaccines adverse reactions compensation system. So that is about the progress of the project. In this project, we pay attention to the following key points. And all these points are also of a lot of attention to the Bill and Melinda Gates Foundation. First, the biggest challenge in the transformation of traditional thinking to international thinking is to change the regulatory thinking and philosophy of regulators. For example, if you go to another country, you can see things are very good there, but we may not be quickly adaptable to the lifestyles there. So there is the process of adaptation. Second, the regulatory system will be reshaped again. And drug regulatory agencies will have the opportunity to be repositioned. Days ago, I think you have seen the State Council document on comprehensively strengthening pharmaceutical or medicinal products regulation capacity. Thirdly, the regulatory follow-suit strategy is challenged. Previously, for domestic laws and regulations or guidelines, we refer to the advanced practices of other developed countries, other countries that are developed. However, the regulatory framework of China has not been internationalized, not internationally recognized. So fourthly, drug regulation has not been truly internationalized, and the international recognition of regulatory results is not high, especially in the aspects of review, inspection, and pharmacovigilance. And fifthly, now I would like to share with you the research we have conducted in light of the problems. The first one is the vaccine marketing authorization holder system research. For example, what conditions should be met for the holder to have the manufacturing capacity? And if it entrusts somebody else to do the manufacturing, if it is different from the manufacturing practices of China, then how to make sure that it is consistent with international practices? For example, change management. In particular, for the vaccine marketing authorization holder manufacturing site change, we conducted a research. Do we need to issue a license for cross-province and cross-region regulation? About emergency use authorization, how can we use it to accelerate the marketing of vaccines and better the management? And second, while encouraging vaccine innovations, we have also conducted research on the clinical trial design and surrogate endpoints used in vaccine innovations. For example, the clinical endpoint of HPV 9-valent vaccine was modified from CIN2 plus to 6 months of persistent infection. We use CIN2 plus and persistent infection as two endpoints. We conducted research on the scientific basis and its predictability of correlation. If we use surrogate endpoint, it can greatly accelerate the marketing of vaccines. But there are also some regulatory considerations to take into account. For HPV vaccines and COVID-19 vaccines, we conducted research on primary and secondary endpoints. And thirdly, we have also conducted a long-term project. It was initiated prior to the COVID-19 epidemic. That is the compensation system for abnormal vaccine reactions. In Article 6 of the vaccine management law, there are now big changes. The state shall practice a system of compensation for abnormal reactions to vaccination. If the death, serious disability, or organ and tissue injury of the vaccinated person occurs during or after vaccination, which is an abnormal reaction to vaccination or cannot be ruled out to be so, then compensation shall be given. The compensation scope is managed by catalog and adjusted dynamically according to the actual situation. This is an article we suggested that is finally written into the law. And how to formulate the catalog, then the compensation scope, standard, and procedure will be formulated by the State Council. That means the differences at provincial level will be addressed. Of course, this will be of great interest to us in the follow-up research, that is EF definition, compensation scope, and the consistency between compensation scope, standard, and procedure. And then is the use of 3R principle in vaccine development and registration. It is widely used in drugs development and registration and manufacturing. Why we should use 3R? It is designed to have animal welfare protection, to be compliant with law, and to do modeling, because with scientific means you cannot have the extrapolation.
就是弱化或者减少的，所以3R 原则呢 ，replacement。
So 3R principles means replacement, reduction, and refinement.
这3R 呢都是基于对动物实验的这种减少来进行，特别是研发阶段。疫苗阶段的应用呢，主要是在临床前的安全性测试以及上市后的质量控制这两个方面。那么可以用一些新的方法
We can use some new methods to replace the original multiple dose tests involving the use of a lot of animals. Internationally, there have been some precedents, and we are also doing such research. In addition to such research, we are also doing some research on regulatory challenges to encourage innovation in vaccine development. For example, regulatory challenges in the development of multivalent multi-vaccines. If you want to manufacture multi-vaccines in another country, at least you must have the capacity to manufacture a single vaccine to produce multiple vaccines. That is in China. However, internationally, that is not required. Therefore, in our closed-door meeting, it was mentioned that there is a partner for manufacturing, each manufacturing different sections for shared manufacturing. So when you are not capable of doing so, but when you are working together, you have the capacity to manufacture multi-vaccines. In this way, the manufacturing threshold can be greatly lowered for multi-vaccines. And in terms of the optimal design of vaccine clinical trials, after 2010, the U.S. has a core section in regulatory science, that is clinical acceleration. That is to have adaptive clinical trial design. During the development of COVID-19 vaccines, we can see in global vaccine network of cooperation, clinical trials adopted a different design. Vaccines for multiple vaccines, clinical trials can be carried out at the same time in multiple countries to complete clinical trials in the shortest possible time period. And in Article 97 of Vaccine Management Law, which encourages vaccine exports, we conducted research on how to encourage vaccine exports to see what difficulties and barriers are still there. For example, what should be approached differently in terms of the legal framework, and how vaccines manufactured in China can be quickly exported to other countries. Whether we should have pre-qualification. For regulatory science think tank project, we have the following main outputs. First, research reports. Second, recommendations by us as a think tank that will be forwarded to government agencies through different channels. For example, Tsinghua University has our think tanks, and Tsinghua University's think tanks can submit the recommendations to government departments. We have drafted multiple recommendations, and some recommendations have been approved by the State Council leadership.
In the future, we will also have monographs. So I have tried to make a brief presentation. Thank you very much to the Gates Foundation, and also I look forward to further further discussion and communication with you. Thank you. Thank you, Professor Yang, for sharing with us many experience related with the regulatory science. I'd like to thank the 4 speakers in this session and 5 keynote speakers. And I would also like to thank our audience online and offline. Now it's time to have the last part of our forum. We will have the panel discussion. Altogether, there will be 6 panelists. May I give the microphone to Dr. Xu Fujie.